after retiring 2 years ago now was to join CONIPS because I wanted to learn the flora and I wanted to establish a native plant garden at my new home in Littleton. And CONIPS programs have delivered really well on both fronts. And so I'm really delighted to be invited to talk to you tonight. In my talk, I want to get rid of this, excuse me. I want to tell you a few stories that I especially like from the history of herbaria. I'll also tell you about how herbaria contribute to my understanding of the environmental challenges we face today. And most he was just talking about. I've been around herbaria all my life. My father was the founder and for nearly 40 years, the curator of the herbarium at San Francisco State University. He poured his considerable curiosity and energy into documenting the fungi and also primarily, but also bryophytes and lichens of California. And I participated happily in my dad's collecting and curating activities until I was a teenager when I wanted nothing more to do with herbarium or my parents for that matter. But in the middle of my college years, I found my way back and I discovered that I too wanted to participate in this great adventure of documenting plant and fungal life and to join my dad and his passionate, often eccentric band of students and colleagues who derive such joy from this work. I arrived at the New York Botanical Garden in 1981 to start a one-year postdoctoral fellowship in the herbarium, and my project was to organize and index the collections of a man named William Mitten, whose herbarium the garden had. Mitten was a 19th century apothecary and self-taught bryologist who obtained specimens from essentially all the famous European exploring expeditions of the 19th and even 18th century. And the work was thrilling but frustrating because often these historic specimens lacked adequate documentation. A scribble written in terrible handwriting, as you can see on those labels, might be all that accompanied a historic specimen. From that experience, though, frustrating as it sometimes was, I developed an enduring interest in botanical exploration and the documentation of historical collections. I managed to stay on at the garden past that postdoctoral years and worked my way up through the administration of the garden's William and Linda Steer herbarium. And among the things that I've loved about working in a herbarium has been the opportunity to visit and get to know the staff of herbaria all over the world. These institutions differ in language and culture, but all are committed to preserving and sharing their wealth of plant knowledge and of plant and fungal specimens. At a time when we seem to be bombarded daily by negative aspects of human nature, herbaria highlight one of our better impulses to save things, not just for ourselves, but also for generations to come. It's true that many times plant and fungal specimens have been, their collection has been fueled by imperialism, and the impulse to kill these organisms just to store them in cabinets may seem to derive from some base hoarding instinct. However, investing time and resources to care for these irreplaceable representatives of past life and sharing them freely with anyone wanting to study them is both generous and wise, because herbarium specimens constitute a unique source of data that help us understand how the world's vegetation has changed over time, and also how to predict how it may change in the future. Evidence suggests that this man, Luca Ghini, created the first herbarium. He was from the region around Bologna, and he lived at a time when the Renaissance was starting to blossom in northern Italy, and forward-looking universities in the region encouraged independent thought, and new ideas and goods came from travelers passing en route from northern Europe to the Mediterranean and the Middle East. After graduating from the University of Bologna with a medical degree, Ghini was hired there as a professor. He offered what is generally considered to be the first botany course offered in the medical school with a focus on medicinal plants. His course was very popular with the students, and within a few years was elevated to a core element of the curriculum. Instead of relying just on classical texts, which was the tradition at the time, Guinea encouraged his students to make independent observations of the plants that he collected for their study. And he maintained these reference specimens as pressed, dried plants glued to the pages of a bound book. Hence, that became the first herbarium. Later, he went on to create the first botanical garden in the city of Pisa when he moved on there to teach at the university. And like his herbarium, the aim of the botanic garden was also to aid in the teaching of botany. Botanists came from all over Europe to visit Guinea, and they adopted the herbarium technique to aid in their plant study. Guinea's herbarium has not survived, but those of some of his students have. And some examples of their specimens are shown here. The one on the right, I'll call attention to. Leonhard Rauwolf was a German botanist who was among the first Europeans to collect in Turkey and the Middle East. And he created the most beautiful of the early herbarium volumes. For reasons that aren't entirely clear, he bordered his pages with a type of wallpaper that was actually designed to cover wooden beams inside a building. As mentioned earlier, herbaria contain not just vascular plants, but also algae, bryophytes, and fungi. And sometimes collections of only fungi are referred to as fungaria. Most of these early 16th century botanists largely ignored these organisms because it wasn't clear until the microscope was invented that how their life histories could be understood. Also, both mushrooms and algae decay very quickly after collection, and they're very difficult to preserve. However, a few bryophytes and lichens and the occasional alga can be found among the pages of those earliest herbaria. In all of these groups, the features that are key for understanding their life history are so small that they can't be viewed with the human eye, which inhibited the understanding of their relationships to other organisms. In fact, because of their inability to see the reproductive structures, they were often referred to as cryptogams, which is Greek for hidden gametes or hidden sex organs. The earliest herbaria that focused on these so-called cryptogams were those of Per Antonio Micheli and Johann Dallenius. Micheli was the first to describe reproduction in fungi, and his herbarium, which also includes that of his students, is maintained at the University of Florence. It's about 19,000 specimens. Dallenius was a German botanist, but hired by Oxford University, and his herbarium at Oxford consists of about 900 specimens. Like Rauwolf, he liked to put a kind of a crazy border on his specimens. At the time that Guinea was inventing the herbarium, other world cultures were building upon centuries of plant knowledge that surpassed that of Europe in volume, longevity, and continuity. This happened in China, in the Islamic world, focused mostly on what is today Iran, and also in India and Mexico. But interestingly, none of these cultures developed the tradition of herbaria. In the seasonal European climate, many plants cannot easily be observed all year long, as we know. However, in other parts of the world with a tradition of written scholarship where the climate is more moderate, plants could be observed for a longer period of the year. So maybe they didn't feel the need to use preserved material to study plants. Also, none of these cultures, but European, had a prolonged period of intellectual stagnation, which was the Middle Ages, which interrupted the tradition of recording natural history information that had been established back by the Greeks and Romans. But when science reemerged in Europe in the Renaissance, the Europeans lacked a knowledge base that had been accruing century by century, which sort of forced them to start over. And the lack of precedent for the knowledge and an intellectual climate that was open to new ideas are thought to have spawned many inventions of the time, such as the telescope, the microscope, and perhaps herbaria and natural history collections in general fall into this category as well. Beginning in the 1600s, European countries invested heavily in sea and land-based exploration, the goal of which was to establish colonial outposts where they could exploit local resources for products for trade and to secure safe and dependable routes for transporting goods back home. Many of the products they were keen to obtain were plants or plant-derived, most notably spices, but also horticultural plants such as tulips, which sold for astronomical prices in the early 1600s. And although the botanical objectives of a government-sponsored exploring expedition usually focused on a few species of commerce, by the beginning of the 18th century, the idea emerged that at least some expeditions should send naturalists to document the native vegetation and animals of foreign lands visited. And these trips resulted in the collection of living plants as well as those preserved as herbarium specimens. Only about 12,000 of the 400,000 known species of plants occur in Europe. So almost every trip brought back species to Europe that no one had ever seen before. And as a result of the influx of exotic flora, understanding the breadth of the world's plant diversity became a central focus of botanical study. The first European explorer charged specifically with the collection of plant specimens for scientific purposes was a sort of unlikely figure, William Dampier. He was a working-class Englishman who started his life at sea shortly after completing his primary education. As a young man, he worked on ships that harvested logwood, a tree in the legume family, along the shores of the Caribbean. And the wood was used for dyeing wool and cloth. But later, he made his way around the world as a privateer, a sort of government-sponsored pirate, sanctioned pirate. And a privateer had permission to attack foreign ships during wartime and to take the contents of the ship as a prize. And the proceeds would be divided between the sponsors, the ship owners, and the captains and crew. So he did that. He went around the world pillaging. He kept a very lucid and detailed written record of his accounts, though, not only his plundering adventures, but also sea currents, landforms, plants, and animals that he encountered. And he went to great lengths to maintain his journal, placing rolls of parchment within hollow bamboo stems, which he sealed the ends of those then with sealing wax. And when he returned to England from this trip, he discovered a new phenomenon that had developed in his absence, which was the coffee house. And an interesting feature of the English coffee house at the time was this is one of the few places where social classes mixed. So Dampier frequented one called Jonathan's in Change Alley, which is still a coffee house, not the same one, obviously. And there he rubbed shoulders with powerful people who were fascinated by his stories, which prompted him to write a book about his travels entitled A New Voyage Around the World. It was hugely popular and it went through 3 printings, and it was translated into Dutch, French, and German. And because of the support, the success of his book and the support of the friends he made at the coffee house, the Royal Navy decided to sponsor a second expedition for him. And he was supposed to survey what they called Terra Australis, which included Australia, but was actually thought to be a much bigger landmass. The English Navy wanted him to explore the eastern coast of this landmass, which they thought extended all the way to the South Pole. And although enthusiasm was high for this voyage, the ship assigned to it was a little disappointing. The HMS Roebuck was a 9-year-old fire ship. And a fire ship is one that, being near the end of its useful life, was slated to be used as a weapon. They would fill it full in the height of a close naval battle. They'd fill it full of combustible contents and then just send it into the enemy fleet, and then it would blow up. So Dampier's expedition departed in 1699. His route took him first to Brazil, as most of these expeditions did. But instead of sailing around Cape Horn, he made the radical decision to sail east, and he sailed farther south than most other ships, taking advantage of these prevailing winds, the roaring 40s. And about 4 months after leaving Brazil, Dampier and his crew reached the western coast of Australia, and they were low on supplies of food and water, but they had lost no crew to scurvy or any other mishaps.
Yeah.
The first landfall was on Dirk Hartog Island in Shark Bay, which you can, if I have a pointer here. Yes, yeah, pointer right about there. And Dampier collected about 20 flowering plant specimens and 1 marine alga at this spot, which he pressed between pages of a book. Although he complained that some of his specimens were spoiled, those that survived were very well preserved and perfectly adequate for scientific study. I think I've got to advance the slide, sorry. At another landfall, he discovered his most spectacular flower, the plant that's now known as Sturt's desert pea. You can see the specimen of it next to the photograph. From Australia, the expedition sailed northwest to New Guinea. And as the trip progressed, however, Dampier became increasingly concerned about the state of his ship, whose hull was badly damaged by shipworms. So he decided not to continue on to the east coast of Australia, but instead to begin the trip home. And the Roebuck made it most of the way back to England, but it sank off the coast of Ascension Island in the southern Atlantic Ocean. They were close enough to shore that the sailors could swim ashore and no one was lost, but many of Dampier's specimens were. And several months later, the crew was rescued by a passing Royal Navy ship, which took them home to England. The surviving botanical specimens include the plants that I mentioned he collected in northwestern Australia and one moss from New Guinea, but not much more. Those specimens were deposited in the herbarium of Oxford University, where they remain today. About 17 new species were described based on his collection, but interestingly, Sturt's desert pea would not be formally described for another century. Dampier wrote a book about his second trip as well, but it was not as popular as the first. And he was dismissed from the Navy because he lost the Roebuck and because of an issue with his second in command, who he feared the whole time was plotting mutiny against him. And although he made many more sea voyages as a privateer, he didn't collect any more plants or write any more books. But interesting to note that Dampier's books contain the first known usages of more than 1,000 words, according to the Oxford English Dictionary, including such commonly used words as barbecue, avocado, chopsticks, and tortillas. The first specimens collected in what is now the United States were probably those of John Bannister, an Oxford-educated chaplain who settled near the mouth of the Appomattox River in what is today Virginia in 1679. He established a parish there and took an interest in the natural history of the region, sending observations and specimens and seeds back to England. John Ray included a list of Bannister's plants in his Historia Plantarum, which interestingly is the same publication that included the descriptions of new species that Dampier had described. And Bannister's botanical career was very short, unfortunately. He was shot and killed by a field companion while collecting plants. The English naturalist Mark Catesby visited Virginia, North Carolina, and South Carolina, and the Caribbean on several trips between 1712 and 1726, making collections and illustrations that form the basis of a book that generated great interest in the flora and fauna of the New World. And it included this very often reproduced painting of Magnolia, which painted by the masterful German artist Georg Ehret. Another early English immigrant plant collector was John Clayton, who served as a clerk for Gloucester County, Virginia for many years and explored rather widely in his free time. Clayton's specimens found their way to the Dutch botanist Jan Gronovius, whose Flora Virginica was published in the mid-1700s, based mostly on collections by Clayton. Linnaeus also studied Clayton's specimens and named the lovely wildflower known as Spring Beauty for him, Claytonia virginica. Clayton's herbarium and of some 700 specimens is now at the British Museum. Although at first interest in the botany of the New World was largely by and for Europeans, by the time of independence, those immigrants who decided to become Americans were beginning to take an interest in developing their own scientific resources, including herbaria. And a lot of that early interest was focused in Philadelphia and the surrounding countryside. Capital of the United States until 1800, Philadelphia was also the first academic center of the country, home to the American Philosophical Society, the nation's first scholarly organization, founded in 1743. The APS was home to the first institutional herbarium in the U.S., incorporating some important early private herbaria as its foundation. Also, Philadelphia was where the first U.S. government exploration that resulted in herbaria was organized, the Lewis and Clark expedition. Oops, forgot to advance. Yeah, the expedition had the goal of exploring the newly acquired Louisiana Territory and open the door to the westward expansion of the country and the displacement, of course, of native people from their land. Meriwether Lewis and William Clark were trained to make botanical expeditions by Philadelphia botanist Benjamin Smith Barton. They were very conscientious in this work and made good quality specimens and excellent detailed field notes. How the specimens were handled after collection was less exemplary. Barton was also supposed to identify the plants when they returned, but he turned that responsibility over to a student of his, a German botanist named Friedrich Persch, who was living in Philadelphia at the time. And Persch identified some of the specimens while he was in the States, but he took about 50 of them when he went back to England, unbeknownst to the Philosophical Society or anyone else. And years later, these specimens that he took to Europe turned up in auction in England. And just by chance, that auction was attended by an American botanist, American lichenologist, actually, Edward Tuckerman of Amherst College, who saw them, recognized what they were, and bought them. And then when he got back to the States, he returned them to the Academy of Natural Sciences, which had in the intervening years taken over the herbarium from the Philosophical Society. For nearly 40 years after that, the Lewis and Clark specimens that Tuckerman bought and brought home were the only ones thought to have existed. All the others were thought to have been lost until about 1896, when they were discovered in a storeroom. And specimens were still bundled in their original papers, and some, actually a lot of them, were badly damaged by beetles. However, despite the lack of interest in these specimens in the years immediately after they're collected, today they are preserved in a manner befitting their national importance. There are 222 of them in all. They've all been digitized, and they're stored in their own room in a climate-controlled space that's designed specifically for them. Oh, sorry, I should have advanced that. Here are some of the Lewis and Clark specimens. An interesting feature of them, you see all the little slips of paper attached to them. These are those are called annotations, and botanists make annotations after they study them. And some of them have 20 or 30 of these little slips on them. So they have a huge amount of importance for, setting a baseline kind of for the American flora. After Lewis and Clark, the US government sent many expeditions to explore the Western US, and the botanical collections from these were handled much more efficiently than Lewis and Clark specimens. Most early on went to John Torrey, and then later to Asa Gray for identification. Torrey was a New York City native who became the first internationally recognized authority on American plants. He planned to write a flora of the entire United States, but he never completed it. He did, however, describe many new species and amassed a herbarium of an estimated 50 to 80,000 specimens, most of which he mounted himself and annotated with his observations and comparisons of species. Among the specimens that he obtained were those of Edwin James, who Coloradans may know was the first person to ascend Pike's Peak. In fact, the peak was first called James Peak until they renamed it for Pike. And down there in the lower left-hand corner is the specimen of Aquilegia cerulea, the first, the collection on which is the basis for that species, the first collection of that species, made by James and named by Torrey. In 1860, when Torrey's collection outgrew its space, he donated it to Columbia College at that time, later Columbia University, and eventually this formed the nucleus of the New York Botanical Gardens herbarium. Asa Gray, who was a student of Torrey's, took over from him as the leading American botanist of the latter half of the 19th century. And his herbarium is the centerpiece of Harvard's herbaria. The Harvard University herbarium today, with an estimated 5 million specimens, is the largest university herbarium in the world. And although Torrey and Gray were undoubtedly the predominant American botanists of the latter half of the 19th century, they were criticized as being what they called cabinet botanists, meaning that they didn't do much field work. They rather relied mostly on herbarium specimens for the species that they described. But although the cabinet botanist designation was largely correct for both men, Asa Gray did make a trip west in 1877. He and his longtime friend, Joseph Dalton Hooker, who was the director of the Royal Botanic Gardens of Kew, arranged to meet in Colorado to examine and collect specimens of the high altitude flora of the Rocky Mountains. This very famous photograph on the left is of the two botanists and other members of the expedition taken at their camp near Levada Pass, located on the border of Postia and Huerfano counties. They were hosted by Dr. Ferdinand Hayden, who was the head of the U.S. Geological Survey and who was leading an extensive survey of Western geology at the time. And to reach Levada Pass, Hooker and Gray traveled on the newly opened Denver and Rio Grande Railroad from Canyon City. This train operated not for a very long time, from 1877 to 1899. The pass is at 10,300 feet, and the site of the camp was supposedly about 300 feet lower. The tents were brought in from Fort Lyon and the other furnishings from Fort Garland. And the expedition camped there for 3 days. Then they went on to Fort Garland, from which they went on a 2-day expedition up Sierra Blanca. The results from their research appeared in Volume 6 of Hayden's Bulletin of the United States Geological and Geographical Survey of the Territories. It was in a paper entitled The Vegetation of the Rocky Mountain and a Comparison with that of Other Parts of the World. And in this article, they make a case for the role of ice ages in providing the environmental pressures that cause species to move from one area to another. It was in San Francisco that the first Center for Botanical Study developed in the West Coast of the United States. And the development of this center created the first real challenge to the East Coast botanical authorities of Gray and Torrey and later ones. The California Academy of Sciences, which was founded in 1853, had the mission of making a thorough, systematic portion of every part of the state and collecting, quotes, her rare and rich productions. The University of California, established a little later, 1868, included the provision for a museum of the university and intended to house the collections made by the California Geological Survey. And the survey, which was instituted to find new natural resources as the gold rush wound down, was charged with documenting and preserving samples of all minerals and plants and animals found in the state. Mary Catherine Lane Brandegee was a central figure in the early years of Western botany. She was born in Tennessee, but moved with her family first to Salt Lake City and then to the Sierra Nevada foothills. Widowed at 30, she pursued her dream of becoming a medical doctor. She enrolled in the University of California, the third woman to matriculate there, obtaining her medical degree in 1878. She became fascinated with plants while studying their medicinal properties. So instead of opening a medical practice, she joined the staff of the California Academy of Sciences, becoming a curator of the Herbarium in 1883. By hiring her as such, the Academy substantiated the assertions that they made in their founding documents. This assertion was, "Be it resolved that we highly approve the aid of females in every department and earnestly invite their cooperation." The California Academy of Sciences therefore became the first institution in the United States and possibly the world to recognize and encourage women scientists, a decision that paid off for that institution in spades. Kate quickly gained a deep mastery of California plants through her extensive collecting in California, Nevada, often traveling by train as far as possible, then walking 20 miles or so per day to obtain specimens. At about 40, she fell insanely in love with fellow botanist Townsend Stith Brandegee, and together they continued the life path both of them had chosen individually, starting this journey by walking 500 miles from San Diego to San Francisco on their honeymoon, collecting plants along the way. So sweet. Though the California Brandegees may have been key in the establishment of a Western botanical community, the success of that community is due largely to the work of Colorado's own Alice Eastwood. She was born in Toronto, but moved to Denver as a young teen. Her love of nature, especially plants, grew during long hikes in the Rockies. She was a talented student, graduating from high school as class valedictorian. However, her family's precarious financial situation made it impossible for her to have higher education beyond that. Instead, she taught high school, but continued to spend as much time as possible exploring the plants of the Rocky Mountains on her own, and she developed a local reputation for her trail experience and plant knowledge. When Alfred Russel Wallace visited Denver in 1877, it was the 28-year-old Eastwood who took this famous tropical explorer, who was then in his 60s, on a 3-day hike to the 14,000-foot summit of Gray's Peak, introducing him to the alpine flora of Colorado. This past summer, I had the opportunity to accompany Denver Botanic Garden botanists Amy Schneider and Lorraine Yeats on a trip to try to find Eastwood and Wallace's route for this trip, recollecting some of the same species that they had collected 150 years earlier. Turns out that finding their exact route is quite a complicated story that Amy is working on unraveling, and hopefully one day we'll be able to tell CONEPs about. Shrewd investing of her modest teacher's salary in real estate generated enough income that by the time Eastwood was 31, she could quit her teaching job and devote herself full-time to botany. She used her newfound freedom to travel to San Francisco to study specimens at the Academy. The Brandegees were impressed with her knowledge and her enthusiasm, and they invited her to assume Kate's position on the Academy staff. A substantial inheritance bequeathed to Townsend Brandegee allowed Kate to give up her position so she could devote herself to collecting full-time, and Alice could be hired as the curator. And she immediately began learning the California flora through the study of existing specimens, as well as extensive field work on foot and horseback through the Sierra Nevada and Coast Ranges. Sometimes she made these trips alone, but she was often in the company of other naturalists. She funded most of this field work herself, and also used her funds to buy collections for the Academy's herbarium. Through her efforts, the Academy became one of the most complete records of Western plant life, and other botanists consulted it frequently. The Brandegees were apparently somewhat negligent curators, and Alice devoted a lot of her time to organizing the collection to make it easier to access. And one of the things that she introduced, which still continues today, is filing what we call type specimens separate from the general collection. Types, every time a botanist describes a new species, they select one herbarium specimen to represent it forever after. That specimen will always be associated with that name, and it is the reference you go to if you were trying to understand what the botanist meant when they described that new species. So these are the most important and most consulted specimens. And Alice had the idea that these should be kept separate from the regular collection so that people could find them easily. She also thought she would store them in a cabinet that was lightweight and movable, so it could be lowered out of a window in case of fire. So those are the ones you'd want to save the most. Eastwood's energy and enthusiasm for the study of plants and her dedication to the Academy's herbarium gained worldwide appreciation through her heroic efforts to save the herbarium during the earthquake and fire in San Francisco on the morning of April 18th, 1906. Eastwood felt the tremor that morning at her home, but since it wasn't strong in her neighborhood, she went to work that morning with no particular anxiety. However, when she reached the Academy, she saw the building was damaged, and worse, the surrounding buildings were on fire. And the marble staircase that led to the 6th floor of the building where the herbarium was housed had collapsed, but Eastwood managed to climb up the banister, which was still intact. And from the herbarium windows, when she got up there, she could see that the fire had was closing in on the buildings from all sides, and she had only minutes to save what she could. And she chose the approximately 1,500 type specimens and her Zeiss lens, as shown in that lower picture. She couldn't lower the cabinet by herself, but she bundled the specimens in a large piece of fabric and lowered them by rope to ground level. And then she hurried back downstairs, at which time the firefighters were desperately trying to clear the area as the fire closed in. And she secured the specimens on a horse-drawn cart and took them to safety. And in a letter published in Science magazine some months later, she wrote, It is a great loss to the scientific world and an irreplaceable loss to California. My own destroyed work I do not lament, for it was a joy to me while I did it, and I can have the same joy in starting again. And that's exactly what she did. Under her care, the new Academy Herbarium grew from a nucleus of those saved type specimens, reaching a total of 300,000 by her 80th birthday. She authored over 300 publications during her career, 200 of these after age 50. At the age of 91, she traveled to Sweden to the International Botanical Congress, where she was made honorary president of the Congress and was seated in Linnaeus's chair. Because of her scientific accomplishment, Eastwood was offered honorary college degrees on several occasions. However, she would not accept these, dismissing the notion that only someone with a title could make significant scientific contributions. Not only did she have little patience for academic conventions, she also flaunted expectations about the appearance and comportment of women. She made special skirts with slits in them so that she could hike and ride horses more easily. And when other women remarked to her that she was able to do things that they could not, she was known to quote Ralph Waldo Emerson in response and say, "Scorn conventions and you always can." She died at the age of 95, and several years later, the Academy named the Alice Eastwood Hall of Botany in her honor. Today, there are approximately 34 herbaria in 180 countries around the world. Collectively, they hold about 400 million specimens. And if you were to spread these out evenly across the earth, there would be about 5 specimens per square mile. But of course, they're not evenly distributed, and many parts of the world still remain poorly collected. Herbaria still serve their original function to document the occurrence of plants and fungi and provide a reference for their identification and characterization. However, recent technological advances have allowed us to broaden the use of herbarium specimens. Understanding the impact of global change on Earth's organisms and ecosystems has become a central focus of biological research today. Documenting such change is tricky because it requires measurements and observations before and after the change event. And herbarium specimens provide one of the few tangible sources of data about how plants and fungi lived before industrialization and through each successive period of technological advance since that time. And because plants are rooted in the ground, they're especially exposed to environmental conditions in the places where they grow. And we can use that to understand a bit about their past. For example, as you know, heavy metals such as cadmium, lead, and mercury have adverse effects on animal life, even in tiny quantities. These elements can reach harmful concentrations in our soil and drinking water as a result of unregulated agricultural and industrial processes, as well as inadequate waste disposal methods. Generally less sensitive to these chemicals than animals, plants and fungi absorb heavy metals in the water they take in through their roots, and these tissues remain in the plants and then are also in the herbarium specimens. Therefore, we can use specimens to determine the concentration of heavy metals to get a historical perspective on heavy metal pollution in the past. A study at Brown University published in 2017 of herbaceous plants collected in Rhode Island between 1846 and 2015 demonstrated that lead concentration in plant specimens decreased during that period as more effective emission controls were applied to manufacturing plants in the area. And interestingly, lead levels in the early years were also found in specimens from the offshore Block Island, which is 13 miles from the mainland, where there was no significant industry, but that indicating the breadth of the pollution, the area that the pollution affected. In the 1990s, scientists discovered that gene sequences could be recovered from the tissue of dried herbarium specimens. And since that time, the process has been refined to require smaller amounts of tissue, and the success rate has increased even with very old specimens. The one on the right is the oldest at New York that was ever from which DNA was extracted, and that's a specimen from 1835. Until recently, most of the DNA-based research using herbarium specimens contributed to understanding the delimiting one species from another and reconstructing phylogenies or family trees to try to understand how species are related to one another. But now there are new and more powerful tools for sequencing DNA, which is known as next generation sequencing. And these make it possible to sequence all the genes in an organism or characterize its entire genome. And these techniques are now being applied to DNA recovered from herbarium specimens with great success. And surveying the differences in genomes among plants preserved as herbarium specimens allows researchers to understand which genes controlled which traits, traits such as leaf height, leaf size, color, flowering time, and how variation in the expression of these traits have developed in different locations and in different lineages of organisms. And current projects using herbariums in genomic studies includes those that are looking for genes related to the development of nitrogen fixation, drought tolerance, and also traits that make certain species invasive. Specimen digitization is the process of capturing images of specimens, transcribing their data from the label where and when they were collected into a database, and then sharing the results online. And today, there are about 80 million digitized specimens from the world's herbaria that are shared through various data portals. In the United States, much of this digitization work was done between 2010 and the present, thanks to a special funding program from the National Science Foundation. This practice has opened up many new uses for herbarium specimens. For example, the map on the right plots the location of herbarium specimens from Brazil. Overlaying map layers show the distribution of Amazonian forests and the location of forest fires in 2019, which allow a visualization of what plant diversity might have been lost through that environmental catastrophe. When making herbarium specimens of plants, collectors are particularly interested in documenting the structures that contain the diagnostic features needed to distinguish one from the other, most often the flowers. But also leaf buds and fruits. And these structures also indicate stages of the plant's phenology or their seasonal life events. And thus, in addition to using specimens just to identify plants, we can also use them to deduce the timing of their annual growth and reproductive cycles. Because we know when and where each specimen was collected, we can compare phenological change over time and space. And with the volume of digitized herbarium specimens that are now available, it's possible to score large numbers of specimens for their phenological state. And as I'm sure you know, shifts in phenology are one of the commonly predicted effects of global change and are a cause for concern because such shifts lead to a mismatch between pollinators and other organisms with which they're interdependent, pollinators and seed dispersers. Studies using large sets of digitized herbarium specimens have confirmed that these predicted mismatches are indeed happening, source of great concern. Estimates suggest that up to 50% of the still undescribed species of plants may actually already be represented in herbaria, but are either misidentified or are in the unidentified backlog that almost every herbarium holds. That's the backroom, cold room, storage backlog at the New York Botanical Garden, representing about 50 years worth of collections. All the ones that could be identified were, but there's still an awful lot and there's fewer specialists. So that room is not chaotic, but it's certainly full. There's great interest at the moment in applying machine learning techniques such as computer vision and data mining to digitized herbarium specimens. This work is at a very early stage of development, but it holds great promise as technology that can build on digitized specimens to broaden the use of herbarium specimens even further. Preliminary attempts that we made in New York to apply machine learning to plant identification from herbarium specimens shows that algorithms can match images of specimens of unknown identity to a reference set at an accuracy rate of between 85 and 90%. And further streamlining of the algorithms and refinement of the processes will likely make the rate of correct matching even higher. So Because of all the digitization and the expansion of research uses, the past decade has been kind of a golden age for herbaria, but still they face both physical and administrative threats. Physical threats being those that lead to collection, to the loss of the collection through outright destruction due to catastrophic events such as flooding, earthquake, fire, or the loss of protective controls, air conditioning, building security. Administrative threats involve decisions that are made by governing bodies to either remove staff support, appropriate space, or reduce the climate control measures for a collection. A study that an intern and I did a few years ago attempted to calculate the risk to the world's herbaria. And our results showed that we identified 10 risk factors, and 65% of all herbaria faced at least 1 to 3 of those threat factors, and 35% face more than 5. So there's a lot to worry about. We've built this magnificent resource, but it is not secure. Yet every year, at least 60 to 90 new herbaria are registered, and the number of specimens increases each year by about 1 million. And governments around the world continue to fund digitization projects. So hopefully researchers will continue to find more novel uses for these specimens and the data derived from them. And with this slide, I think Alice would be proud to know that the Herbaria of Colorado have grown to nearly 18 with nearly 1 million specimens. Herbarias are all listed here. I can't vouch for the accuracy of the numbers, but that is from the Index Herbariorum, which is the index to all the herbaria. Herbaria benefit greatly from volunteer labor. So if you're interested, I hope you'll consider volunteering at your local herbarium. I volunteer at the Catherine Kalmbach Herbarium, and I really enjoy not just the work, but the companionship of my fellow volunteers from whom I've learned so much. Okay, thank you very much for your attention.
Oh, I just noticed the last graph. That's cool.
Yeah. Those are specimens. They weren't prepared that way, but one of my staff at New York combed the herbarium for specimens that looked like each letter of the alphabet. And then she made an alphabet of them, and then we could pick from those to spell words.
They do look like the letters.
Yeah, she did a good job.
I had a question.
Wow.
How much tissue material is needed to do a DNA sequencing?
I think we would say to people that they could take a 1 centimeter square piece of tissue, and we must have based that on what people told us they needed. So it's not very much. The problem becomes if not just one person does it, but a whole lot of people do it. So most herbaria will allow, we call it destructive sampling, but they do limit the amount. And for some reason, we settled on 1 square centimeter, and that seems to be okay. The ideal thing is after people sequence DNA, they put the sequences in GenBank, which is the repository for those data, and other people don't need to do it again. However, techniques change, people look for different genes, and so it's not at all uncommon for a specimen to be sampled multiple times.
Speaking about techniques changing, has digitization has improved over the years.
Oh yeah. Oh my God, yeah.
Is there any need to go back to some of those earlier digitized records and rescan them with the better technology?
Quality? Yeah. So yes, the first ones that we did, I think, were we paid $20,000 for our first camera back in around 1990, and the pictures it captures aren't nearly as good as on your cell phone. So we did in fact redo all of those. Okay. 600 DPI is sort of the standard. However, for some herbaria, that's an awful lot of storage, an awful lot of to be stored. However, if we're going to do more things with these, especially AI, the higher resolution is really necessary.
Mhm.
So in the end, the best decision is to use the highest resolution available and do what you can. Because the other alternative is at one point you probably will have to redo it.
Mhm.
So it sounds like this is totally different than like back in the 70s or something. It was chemicals they used to separate the chromosomes to look at. You weren't really, and then using the electron microscope, it wasn't.
Right, that sort of chromatography approach.
Yeah.
Yeah, no, they don't do it that way anymore. Although in some ways these rough plates that they get remind you a bit of the old fashioned chromatograph or whatever. Yeah. But yeah, it's in next gen sequencing. I don't really understand that at all. But they can get a huge amount of sequence. In fact, herbarium specimens are even easier to use than living plant material, because when you do these whole genome sequences, the first things you do with the tissue is break up all the DNA into little tiny pieces. And in the dried plants, it's already broken up. So you kind of skip a step. So you get great results. So yeah.
And the enzymes are probably dead too.
Yeah. Yeah. Sure.
So that, I mean, that's a big problem with the DNA sequencing
Mhm.
is that you have enzymes that may start breaking things down.
Right.
Before you you have everything stabilized.
Right.
Did you say, sorry,
Go ahead.
did you say that 60 to 90 new herbaria?
A year.
created a
Yeah.
year?
Yeah. I know that because for the past 20 years, I've been editing this. It's called Index Herbariorum. It's an online index. It used to be books. It's actually started back in the 1930s. They made a list of the herbaria and they just kept adding to it. And my boss before me was, in charge of it. And when she retired, she handed it on to me and then we put it online. So it's called Index Herbariorum. And with that, people can both update the information about the herbarium and they can register a new herbarium. And so I keep very careful statistics and I do an annual report. And last year was it 2021, but one of them was close to 100 new herbaria, which was amazing. But I've never had lower than 60. Now, most of these are in other countries. Brazil always accounts for a number of those, Asia, Southeast Asia, India especially, but there's always at least 15 or 20 in the United States. They might be national parks or other kinds of lands like that, but they are often small universities. There are a few private companies that have herbaria. Amway has a herbarium, as does the company that makes Tabasco sauce, McElhenney company down there off the coast of Louisiana. They have a herbarium.
And do most of these have government funding, at least in the States?
No. I mean, they may have, so there are a number of government herbaria, so those are fully funded. Universities support quite a few. The largest number of herbaria are associated with universities and colleges. So in a way, they're government funded because they're funded by state or local, governments and then there are like the institution I worked at and several others, and like the Denver Denver, I'm not quite sure, but at least some of the other big ones are private, just private institutions who do get government funding in one form or another. But National Science Foundation makes some grants available, but it's mostly for herbaria that are kind of in trouble, that need some kind of rehousing or a repair or something like that. So there's some money available, but not a whole lot. The Institute for Museum and Library Services also provides some funding. But funding is the number one, herbarium curators will always tell you that funding is their number one concern. But I mean, if this project taught me anything, I really don't think there's ever been a time when herbaria were completely secure and well-funded. There are still a backlog of specimens at the British Museum and in Paris, which is also a very old herbarium, specimens collected on these early exploring expeditions from the 1700s that have never been identified. Nobody ever got around to it. So there's never been if there's been a golden age, it's been in recent years, not not years ago, always kind of struggled, you know. Because it takes an incredible amount of time, just time and labor, you know, to preserve these.
No, my God.
Yeah.
Oh, sorry.
Oh, yeah, I was just gonna say, you mentioned like computer vision. Is there anybody working on a program right now or?
There are a
Wait,
couple,
and let me
okay.
ask a question from the
Yeah.
chat that goes right on with that.
Yeah.
Do you know of any projects that utilize 3D scanning in the field to assist with image plants? In addition to collecting specimen for herbaria?
3D scanning, I know has been used for seeds. Especially very small seeds. Maybe fruits as well. Plants, I mean, certainly not herbarium specimens. They're already kind of two-dimensional, so there wouldn't be a whole lot of point in that. But 3D scanning of plants, I assume that probably does go on, but it's kind of not, would be more in the realm of living, you know, documenting living plants. I do know there's a lot of scanning of 3D scanning of other types of natural history specimens, bones and fossils and things like that. But I don't know of a lot of plants. Plants lend themselves very well to the kind of flat picture taking that we've always done. So maybe there's just not a lot of.
What did you say?
I was just wondering if you knew of any kind of computer vision or AI.
Well,
that's going on right
yeah, so they're working on this at Smithsonian has probably done more in terms of like actual results. I mean, they used it, they did 2 pilot studies. They tried to tell apart 2 closely related species of I guess Lycopodium or Selaginella, one of those groups. I forget what the exact nature of the study was, but they tried to teach it to identify these 2, which I guess are I don't remember the 2 species, but look pretty close, but can be discerned using herbarium specimens. And then they did another one to look for paper, not even anything to do with the specimens, but to look for paper that had staining on it that was indicative of preserving with mercuric chloride, which used to be used as an insect, you know, to kill insects, but it's highly toxic. And so what they were hoping was they could, and it makes very characteristic brown blotches on the paper. It looks a little bit like someone dripped brown or like gray water on the specimens, or a dilute gray ink. So they wanted to find those, and then they could kind of remove them and keep them in a special facility. And I mean, whether they found all of them or not, I don't know, but they found them. But what we were working on at New York was just whether we could take knowns and compare them to unknowns and how well it would do.
Yeah.
And we're pretty pleased with the success, but I don't know that they've taken it to the next step and made it, you know, what you'd love is like to just be able to run thousands of specimens through this just by pushing a button from a portal. And we're not nearly there yet.
Are there any Zooniverse projects?
Yes. In fact, early on, what's this? Probably about 2010, there was something called Notes from Nature, which it's still going on, actually. I should say it's not over, where Herbaria would digitize their specimens and put the images up there, and then people could transcribe. They get volunteers to transcribe the data for them. And it worked great. We did it quite a bit. What we found was that people had a hard time, you know, with reading handwriting, especially of taxonomic names, because, you know, the word isn't familiar to you and you don't really know. But so right. So what we mostly had people do, just to help us a little bit, was to identify the state or country where something was collected, and then we could group those, and then we could more easily, you know, complete it. But that's still going on. In fact, there's a project called We Dig Bio, and every year they have a big event, and they try to get people, herbaria all around the world, to do a volunteer digitizing event. And so yeah, that's still very much a thing. And it's interesting. You can get, it's like almost all of these projects, you get hundreds of people who try once, and then you maybe get 5 who get kind of obsessed with it, and they just do hundreds. They just sit there and they just.
Oh, that leads to a question in the chat.
Yeah.
And the personalities of our members. How do citizen science reports like iNaturalist affect collecting and evaluating the presence of species in an area that may not have herbarium specimen vouchers?
Well, I mean, it's clearly really, really important. And there's been a lot of linkages now, they're beginning to be between iNaturalist and, various collections. So that you can, when you search for collections, you can see not only what specimens have been collected, but what iNat observations occur there. And that's really important because I think it's only with iNaturalist observations we're ever going to get at what's missing. Herbarium specimens only tell you what's there, and observations only tell you what's there, but observations over time, people go out every year, and stop seeing something, you'll have a much better sense. And observations are going to become more and more important because we can't just keep indiscriminately collecting and making herbarium specimens forever and ever and ever. I mean, there's a need to keep documenting the flora as climate changes for sure, but their space is not unlimited. Resources are going to become more limited. And maybe, we'll have to depend more and more on observations to tell us what's there. There's always that danger though, that, the observation is ambiguous and there's no way to go back and check it. But for most common things, that's not necessary. So I think a really important thing we should be doing is incorporating the two much more closely.
Able to key it out, because what is she recalling in
Right,
your choicing?
right.
And so we would just toil over like a single plant, for an hour, but it was always super interesting. We learned a lot.
Yeah.
And we got sensitive to the difference between like an aesthetic photo and a biologically useful one.
Yeah. Yeah.
And we do have one more question here.
Sure.
What is the nature of any degradation of pressed samples over decades, centuries? Like, how does a sample age and how does it affect its scientific value?
It's a great question. Well, they get browner and browner and they get more brittle. But what's even worse, the paper they're attached to gets more brittle. And often the biggest problem with an old specimen is not the plant, it's the paper it's on. And you don't want to touch it, because it might break apart. But if, curation has been kept up and the plants have been moved to a new piece of paper or had a backing put on them, the plant material lasts a really long time. I've looked at some of those ancient herbaria that I showed a picture of, and I mean, they're not great specimens by anybody's measure, but you could still see a fair amount of detail. I don't know what as I say, the oldest that we've gotten DNA out of is from 1830s. But as long as they're kept dry, they aren't eaten by insects, so they don't get moldy, and the paper doesn't disintegrate, they're incredibly long lasting. They have to be kept out of the light. Light will destroy them too. But that's the main thing you invest in a herbarium is all the climate control so that you can keep those degradations from happening. But no, the parts of the flower in position to one another, which is key for identification, will be there. The leaves, leaf venation, the shapes and sizes are largely intact.
Hairs,
Sure, yeah, you can see hairs in those ancient specimens. I mean, those could be rubbed off if people have been careless with them.
right?
But the thing about some of those really early volumes, they were actually meant as gifts for kings and queens. Raoul herbarium, the one I showed you with the border wallpaper, that was actually presented to the King of Sweden, who kept it for many years and then it eventually found its way to the museum in the Netherlands. But they weren't heavily used. They were beautiful items and someone took them out and looked every now and then. But those specimens, and those were put on 100% cotton rag, they're beautifully preserved. The hardest ones are probably from the 1840s to 1870s or so, where the paper was more brittle and where they were used more. Those are probably the specimens in the worst shape. But it's not because of the plant material degrading, it's because of how they've been treated.
I actually have a follow-up question to that. And so I've noticed that, people use just any old paper towels or newspaper or whatever to press them. Whereas with art, you're always
Yeah.
you know, this whole thing of acid-free paper,
Sure.
acid-free paper.
Yeah, yeah.
Is that an issue, or is it, you know, that it's really
Yeah.
not, they're not in contact with that paper for that long?
Well, sometimes they are. I mean, we have many specimens at New York in the storeroom that have been in contact with cheap newsprint for 50+ years.
Okay.
Yeah, because they've never been mounted, because they've never been identified. So, you know, of course, when we glue them on paper, we use absolutely archival material. We put them in folders that are archival. I mean, this all is extremely expensive. But we do take those, but when they're collected, you know, newspapers are what you use, because you can always get yesterday's newspaper anywhere in the world. That's probably changing. It's probably getting a lot harder now. But when you go on an expedition, you do not want to take paper with you along with all of your other gear. So you use what you can get, but it's terrible for the plants. I mean, yeah. But there isn't really a great alternative. So that's how it's been done. But if you had unlimited expenses and someone to carry things for you, having pressing papers made of cotton rag would be ideal. but not practical.
Make sure that's in your Sherpa's pack.
Yeah. Thousands of sheets of paper.
Okay, so question, how do plant scientists navigate extracting rare plants in the wild for herbarium? What rules do they use for when it is okay or not okay to extract?
Right. I imagine I would hope that nobody anymore would just collect all of a population of a rare plant. I mean, I can't imagine that happening, but I know it used to happen. The much bigger problem in that area, though, is people who collect things for horticulture or even collections, cacti especially. People dig up cacti left and right. But I know I was just in the field with botanists from DBG, and we were collecting in Sedgwick County, mostly because very few collections from there. And we were really collecting along the roadside. So we were not collecting rare and endangered species. We were mostly collecting the common things. But even in those cases, unless there were 3 different, populations, they didn't collect one, even if it was a pretty common grass or, roadside. So they're very, very intentional about not ever in one site collecting all of something. And if indeed sometimes, even if there was plenty, but still really, not a terribly common plant, they would certainly not take the roots. They would just take a clipping. A good botanical specimen technically includes the roots. So you do basically remove plant material from it. But sometimes you can get away with just a snippet of a stem and not do that. I would hope there is no sort of written code of ethics that botanists should follow. But I think everyone these days is very conscious of not cutting off our nose to spite our faces. You can't at the one time be claiming the purpose of all of this is plant conservation and then be digging up plants. But there's another side to collecting, the dirty side of it. People want stuff. They want to collect something. They want to have it. And especially if it's really pretty, they want to have it, and being rare too. So that's kind of the impulse that has to be pushed back on. And I think I'm reasonably confident that most people do that.
CONUPS does have a code of ethics
Good.
for plant collection that's listed on the website.
That's excellent.
And everyone is supposed to acknowledge it for a field trip.
Everyone who goes on a field trip is supposed to. Oh, good.
And these issues are arising for us because we're doing seed collecting with the Forest Service. So, thinking about how big is the big population,
Sure.
what is good enough?
Yeah.
And then if we have the opportunity, this is the one thing that bobbles around is like, oh, you come across something you don't see very often, like you'd want to collect some seeds, so you'd have some. But then you're never probably going to get to the 3 population rule if you're trying to collect forbs for seed collection. Yeah.
The question of what is enough is one that really needs to be addressed. I mean, how do you know if you have enough? What would be adequate coverage, at a given time of a given species? I mean, one from every county, or one in flower, one in fruit, and one not. I mean, but there are no standards about that. We've never talked about it. It's mostly, we just want more and more and more. And I really think we'd be very smart to think about that in terms of biology and conservation, both of the plants, but also just conservation of space for herbaria, which is not unlimited, you know. But no one wants to talk about that.
Well, then how much is enough over time?
Yeah, and how much is, and is every year, you know? And does it depend? I mean, if it's a common species, is that important? You know, these are like really hard kind of biological questions that, you know, we've not put our minds to.
Gives you something else to do for all of us.
Yeah. What constitutes complete knowledge of the plants of a given area?
I think the like chemical analysis is really interesting, not just DNA, but
Mhm.
the heavy metal uptake, because here we have a lot of mine reclamation lands, and
Right.
you know, it would be interesting to look at some of those high elevation herbaria specimen and
Yeah.
see if they're different in a pre-mining versus post-mining environment.
Yeah.
Because bioremediation is like critical, I feel like,
Yeah.
to part of this is to
Mhm.
that practical application.
Yeah, absolutely. Yeah, those studies should be done more. I mean, it's true, because I think, you know, the Rhode Island study showed that beyond the area where you could see the effects of pollution, the plants were definitely still taking up heavy metals.
Mhm.
So it's a wider range than you think. Of course, this is different. This is mining in the ground, not in the air. It's not so much in the air, I guess, but still.
Big downstream.
Yeah, downstream, absolutely. Yeah, downstream.